Good morning, everybody. Morning, folks. Hi, everyone.
Good morning, Mr. Kidusol.
Hey, how's it going, Matthew? Good morning, Jessica. Good morning, Colin. Morning, Sahil. Morning, folks. We got a nice little day planned for today. Good morning, Maha. Good morning, Olivia. For the lesson sheets are the slides that go with it. Are there slides? No, it's just the filled in form. I'm going to use the filled in notes to teach these lessons for this unit. The slides I didn't really ever make. I just filled them in class. The problem with this is that I never made slides for this unit. I started with unit 1 the last time I taught this. And then I was like, or 2 years ago, I guess. And then I was like, these slides are such a pain because then I end up filling in the note with you all anyways. And so it's like double work. And then people are like, where's the filled in note? And then I was just like, all right, I'm just going to do the filled in note. So as I recall back to my earlier experiences teaching this class, that was kind of my thought process. So yeah, we start our new unit today, proper new unit. And yeah, so it's going to be a little exciting in the sense that we get a full new unit. We start with genetics. And it's going to be exceptionally exciting. So I'll start pulling that note up as more people join. And hopefully we can get that critical mass in the next couple of minutes. And we will get started quickly because I don't want to waste too much time on account of we got stuff to do today. Of course, I've zoomed out to do your interim reports. There we go. Nope, too much. There we go. Alrighty, so let me get our profile and our folder out. Ooh, slow today. There we go. Alrighty, so I've heard your demands in multiple classes. Ooh. And I've decided. Let me get, hold on, I gotta get my computer's so far away. I have my dual monitor situation. I gotta adjust, there things go. Okay, so I've decided I've updated your outline, folks, for those of you that we were talking about a little yesterday, and I can't remember who it was. Maybe it was Anna, maybe it was Ava, maybe it was Rose. Actually, I think it was Rose. Rose, was it you? Is Rose here right now? Was it you that was like, yes. I was like, I remember it was one of the 4-letter name people, and I was like, it wasn't Anna, it wasn't Dean, and then as I was going through my list, I was like, oh, it's Rose. Anyways, Rose brought to my attention that our test was going to be on our April break. So because of the nature of our course schedule and because of the nature of kind of everything that's going on around the time that I wanted to assess you, it didn't really feel fair for me to kind of just chuck an assessment like 3 weeks after we finished the unit. So I'm not going to have a final test for this unit. There will be no final test for this unit, but I've modified the final summative, the problem set for this unit, and I'll give you more information as time goes. Wait, this isn't my updated one, is it? Hold on 1 second. I've updated it, and now I'm like, where's my updated one? Hold on a second. Let me see. Yeah, this is the one. Okay. Oh, wait, why is this not updated? What the heck? Oh, yeah. Okay, it is. All right. Yeah. So then with the problem set, which I'll talk more about as I assign it to you and we go over it, you're basically going to have to explain things to me, record yourself explaining things to me with regards to that problem set. And that will kind of be like your final summative activity. But you'll see that there's going to be plenty of time for us to do it. It won't be due for quite some time. As you see in the schedule, the final explanations, which I'll talk more about later, are not going to be due until April 23rd, but that buys us an extra week. And for some of you who would like working over your break, you can always do some of that over the break. You don't have to. I'm giving you time to do it in class or for homework. But those of you who like to do stuff over the break, you have that opportunity to do so. But again, I'll explain more next week or 2 weeks from now, I guess, when I assign the assessment, because I'm quite excited for it. I think it will streamline a lot of things for us. For me, especially when it comes to some marking, I think it will make it very, very interesting for those of you who, who find that they learn better when they talk. I think it'll be really interesting for those of you who don't really have that skill for you to develop it and start developing it. So I'm very excited for that final summative, and I will, like I said, I will explain more as time goes, because I think it will evolve, so to speak, over the next couple of days and weeks while I continue to build the assessment or add on to the assessment a little bit relative to what I've done so far. And then once I have it developed fully and it is the day for that it will be assigned, I will assign it, and then we'll go into more detail with regards to that. But again, those of you just joining, there will be no final test for this unit. It will just be a final summative that I hope will be a little bit of fun. And then we'll see if I can't build off of that type of style of assignment for the next coming units. That way it makes things a little bit more interesting, a little bit more. My big thing with this problem set is usually what happens in day school, and it probably would happen here as well. But usually kids get together, they all work on it together, and that's fine. But a lot of the times, the kids don't actually know how to do the actual questions because their pal did it, and then they kind of copied it, but not really. And so it's like, fine, you did it, but I want to make you understand it, right? The doing it isn't the biggest thing. It's the understanding and comprehending and connecting. And I think a lot of that will come from the parts where you have to explain it. But we'll cross that bridge later. As we get more people coming in, I just want to make sure that we get everyone down. And once we get this, we can get started. So let me just take attendance real quick. Missing a couple of people. I just want to see whom we are missing. Vicky, Valeria, Talia, Sophia, Stanley, Sarah, Sahil, Rose, Rocco, Ritika, Olivia, Matthew, Mateo, Maria, Maha, Krish, Katie, Jessica, Fayang, Emma, Dean, Colin, Cole. No, Cole. Okay, that's who it is. Sometimes I start at the bottom of my list. Oh, there's Cole. And yeah, wait, who are we missing? We're still missing one person. Amina, Anna, Ava, Carson, Colin, Cole, Danielle. Ah, man, found him. All righty. All right, folks, it's 9:10. We've waited the extra 5 minutes. I got to put socks on. My feet are cold. I don't know if anyone else is. It's a beautiful day out today, folks. My hope is that you get outside at some point, but my feet are always cold. I don't know if that's a thing with people or not. Doesn't matter if it's hot, doesn't matter if it's cold, summer, winter, I always have a pair of socks on because I get cold otherwise, and I don't know why. Alrighty. Same. Yeah, I don't know. It's weird. I'm usually a warmer person, but when it comes to socks, I need a pair of socks on. Doesn't matter. Doesn't matter. I never take socks off. Well, yes, I think I'm very similar in that regard. Alrighty, folks, let's get started to lesson 1 of our brand new unit, genetics. So in this unit, we are going to cover many different topics relating to genetics, genetic processes, reproduction, and how genes are inherited, passed on, and everything in between. This is maybe one of my favorite units. I remember very, very strongly and very vividly being an undergraduate and studying genetics and proteomics, microbiology, molecular biology, cellular biology. And of all of those classes, the genetics and genomics and proteomics classes were my absolute favorite. Chemical or biochemical processes with regards to genes, genetics, genomes, and everything of the sort are an incredibly, incredibly fascinating, brand new science, for lack of a better word, because we've only really understood it over the last 20 or 30 years, right? When you think about physics as a whole, as a science, we've been studying it for hundreds of years, maybe even thousands of years. whereas biology is still a newer science. And even though we have been studying certain things with regards to biology over the last couple hundred years, the idea of genetics and heritability is one of the newer sciences of the harder sciences. So it's a fascinating new field. We're still learning such an enormous amount with regards to genetics. And this is your first step for your understandings of the pure biology that is genetics and heredity. So I'm excited to share that with you. I'm excited to have you learn this new material. If you've learned something like this before in one of your other classes, like your social science classes, that's awesome that you have a nice little introduction, but we really get into some of the aspects with regards to genetics and heritability. And then next year, you look at the biochemical processes, which are even more fascinating of genetics. So without further ado, let's get into it because it is a shorter lesson, but I want to spend some time talking about a lot of the basics because there is a bit of information still. So with regards to, am I recording? Let me just double check that I'm recording. Oh, yes. Okay. Sorry. I was like, am I recording? All right. So with regards to genetics, specifically heredity, what is the whole point of cellular division? Why do cells divide? You looked at your review for mitosis yesterday. My hope is that you did that self-quiz and you kept your results handy for when you do it again on Wednesday. But what is heritability? Well, the idea of genetics or chromosomal information being passed on, it needs to happen in order for that cellular reproduction because cells will divide for growth, repair, and like I said, with regards to reproduction, which is going to be the main focus that we look at in this unit. The chromosomal information or our genetic information needs to be passed on for those instructions to continue to be followed by that cell. A cell uses its DNA or its genetic code to be told the instructions that it needs to do in order to carry out functions for life. So the idea of passing on chromosomes and traits is known as heredity. Heredity or genetics as a whole is that study of how information or how that chromosomal information is passed on, as well as that variation in the inherited characteristics. We have a couple of activities that we will look at in this unit as well as next unit that kind of looks at the overall idea of how traits are passed on. You're going to look at your family or your immediate family, parents, siblings, if you're lucky or you have the opportunity to, grandparents, nieces, nephews, cousins, anything in between to kind of compare those observable traits that tend to be very, 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 very consistent within a family. So we're going to spend a bit of time looking at that, and we're going to have some fun with it. Alrighty, so let me just All right, questions so far? Chat. What counts as a trait? A trait is something like blue eyes, gray hair, or brown hair, height, in some aspects and in some ways, personality, intelligence. Those are all traits. Those are all traits. No, all sorts of different traits get passed on. So we kind of talked about it last unit, right? We kind of talked about it last unit with regards to your mitochondrial DNA, right? That mitochondrial DNA gets inherited from your mother. So even something like the mitochondria within your cells are an inherited trait that you get passed on with regards to your mother. If you are a male, then your genetic information that makes you a male, that Y chromosome, which we will spend some time talking about, that is inherited from your father. You cannot inherit that from your mother, right? Your biological father gives you that Y chromosome. And so those are two examples of things that are passed on or inherited traits that maybe not necessarily are like skin color, eye color, hair color, or height, or whatever. And the way that all of that inherited traits get passed on is through what's called genetic material. So genetic material is the idea that we're going to talk about in terms of how an organism stores that code or stores that genetic information in order to pass it on. and it's stored in the way of DNA, or deoxyribonucleic acid. Now, many of you have probably heard this term before. Many of you know what this term means, but DNA, or deoxyribonucleic acid, stores information in the form of chromosomes. These chromosomes are groups of genes that are responsible for passing on our traits, so to speak. So humans typically have 46 chromosomes. They are identical chromosomes within every single cell, every single skin cell, every single brain cell, every single muscle cell, stomach cell, bone cell, any cell you can think of, with the exception of your reproductive cells, has the exact same and identical DNA. So this DNA is in two sets of 23. Now, you might see this two sets of 23 come up often in this unit. We talked a little bit about it last unit as well. So we have 46 diploid number, 46 chromosomes. That's our diploid number. Two sets of 23. Okay, I'm going to spend more time talking about what that means as we move through meiosis in our next couple of lessons, as well as mitosis. But the general idea here is that you get one set from each parent, right? One set from each parent. So two sets of 23, 46 total. 46 is our diploid number. If you were to say, oh, what is our haploid number? What's our haploid number with regards to our chromosome information? So a human being, diploid 46. Our haploid number is 23. Yes, thank you, Jessica. Because remember, diploid, two pairs or two sets, haploid, half of 46, 23. And we'll spend some more time talking about what that means, how that gets, how we calculate that. I mean, it's just half, but in different species, you have different haploid and diploid numbers. So it'll be important for us to consider that as we go. All right. So with regards to the structure of DNA, each chromosome, on each chromosome, there are genes. These genes are specific sequences that are going to tell the cell how to function, okay? So it will give an organism its trait. So each gene is located at a specific location known as a locus, or a loci, or loci for an individual, or sorry, for plural. Now, in the past unit, we wrote 2n as 46. I believe, what does the n represent? Yeah, the n represents one complete set, Jessica. So one complete set. So one complete set is n, or the haploid number for any species. But I will spend some more time talking about that in this unit, I believe, or this lesson, I believe, sorry. If not this lesson, then next lesson for sure. But I will spend some time talking about what n represents as we move through mitosis and meiosis. But yeah, no worries, no worries. So yeah, these genes are located at a locus or a loci, and each chromosome has hundreds, if not thousands of genes. Now again, right, we have 46 total chromosomes, okay, two sets of 23. On each of those chromosomes, we have hundreds, if not thousands of genes that are responsible for giving us our traits, our behaviors, in some way, how our body functions, and overall, how we are as a species, really and truly. Just adjust there. Okay. So when we talk about some of the specifics with regards to the chromosome structure, the centromere is this part in the middle here. It is responsible for connecting two sets of copied chromosomes together. When I spend more time talking about mitosis, the centromere will come up more often. I just wanted to draw your attention to that centromere here because it will be an attachment point when we look at cellular division in mitosis tomorrow. Some of you reviewed it a little bit yesterday, as well as from last year, and then that quiz that I made you do yesterday as well. But the centromere is that point where the cell will attach itself to help with regards to separating that copied DNA. All right, are there any questions? Any questions as of yet? me just finish doing attendance. All right. No questions I see. All right. Oh, there are questions. What's the word beside during? 1 second. Let me see. What does the text say to the right? So during S phase, I'll type that in. I'll type that in. The one in front of cell cycle. Yeah. So it says during S phase, and then in brackets, cell cycle. That's what y'all were asking about, right? If I'm not mistaken. Yeah. S phase is from mitosis. We'll spend some more time talking about it tomorrow if you're not sure. That was from your grade 10 review. And if you're not sure, we go over it again tomorrow. So if you don't really remember, that's okay. We'll spend some more time talking about it tomorrow. Yeah, no worries. All right, let's take a closer look at, oh, bother. There we go. Let's take a closer look at the chemical structure of DNA and some of the historical concepts that we learned with regards to DNA as a whole. So the idea of X-ray crystallography, which you don't really have to know what it is, but ultimately it is the idea that you can take a picture through a crystal using X-rays to see structures of microscopic things. And scientists discovered that the arrangement of DNA kind of forms this helical or double helical spherical structure, spiral structure. And Rosalind Franklin was one of the pioneering scientists that did it in 1952. So she used this crystal with this, with this cell in there, this bacterial cell, and she shot X-rays through it, and the crystal kind of magnified some of the structures within that bacterial cell. And she found that it is a spherical or helical structure. And then Watson and Crick modeled DNA into a double helix later that consists of a sugar phosphate backbone as well as nitrogenous bases. So I have here, just as the image you see to your left is a DNA X-ray crystallography picture. And as a result of those important studies by Dr. Franklin as well as Dr. Watson and Crick, we were able to model the general structure of DNA to kind of get a good idea of what makes it up. And so when I talk about those nucleotides, right, when I talk about those nucleotides, I'm really talking about the individual subunit pieces of DNA. So nucleotides are the individual subunit or pieces of DNA. So nucleotides are the tiny pieces that make up the DNA strand. So it includes a phosphate group. So again, just kind of getting into some of the things. I can't remember which period it was. I want to say it was period 4. Yes, period 4. One of my students asked, because they're learning about some aspects of organic chemistry in their chemistry class. And she was talking about the connection between chemistry and biology. You'll start to see that connection come to fruition in this unit. A phosphate group, right? This phosphate group is connected to a deoxyribose sugar as well as a nitrogenous base. And so I'll talk about the structure here as well. But ultimately, there are hundreds of thousands of these nucleotides that are bound together over the series of one single chromosome that make up one DNA strand for that chromosome. So millions upon billions of nucleotide sequences make up who you are. So a nucleotide consists of, as I said, that sugar phosphate backbone here. So there's the phosphate group, and then the sugar group, the deoxyribose. For those of you familiar with our chemistry, deoxyribose means it lacks an oxygen on one of the carbon chains. So deoxyribose phosphate backbone, right? That sugar and that phosphate group make up the backbone. And then we have the nucleotide. And this, for our purposes in this class and in this unit, it is the most not important thing per se, But it is the big idea that we will be spending time talking about, because this nucleotide, let me just, this nitrogenous base, this nitrogenous base is what allows for DNA to kind of have that message and kind of do the thing that it needs to do. Just trying to get this, there we go, bam. So this nitrogenous base allows for DNA to kind of function the way that it does. And then again, like I said, with regards to that double helix structure, it is going to consist of 2 strands of those nitrogenous bases, or that DNA strand. And overall, combined together, it makes up that DNA double helix structure. All right, questions? Is there any meaning behind the shapes chosen in the diagram? Yes. Well, no. Yes and no. The only meaning is that deoxyribose, it's a pentose, right? It's got a 5-carbon chain for the deoxyribose. We don't really get into that in this class. You will in chemistry as well as grade 12 biology. But for the sake of this class, we don't get into the biochemistry, because it would knock too many people's socks off, especially those of us who took this class to be like, I don't want to take chemistry. Chemistry's hard. And then all of a sudden I drop chemistry on your head. It's we don't get into it, so to speak, but you will need to know what that is for next year, grade 12 biology. So we do spend a bit of time talking about it in digestion, where we look at the difference between the sugars. But again, it's for the sake of genetics, we don't really get into the specifics with regards to the meaning of the structure of it all. All right, any other questions with regards to DNA as a whole? Okay. I'll just give everyone a second or two to kind of process and digest. And if they have questions, to ask them. I will send off my attendance. All righty, no questions, it seems. We can start to look at the structure of these nitrogenous bases, because this is what allows for that DNA code, or that DNA sequence, to really have that robust amount of information that it can store. Because the nitrogenous bases are made up out of 4 different nitrogen bases for humans. We talk a little bit about uracil when it relates to bacterias, but and RNA specifically. We'll talk about that in a bit. But the main groups of the nitrogenous bases that are making up human DNA are those 4 nitrogenous bases that I do not have highlighted. So guanine, adenine, cytosine, and thymine, all of those nitrogenous bases, you can see how they contain a nitrogen group in each of the structures, and it has to do with the naming of it. Oop, did I miss one in the, no, okay, we're good. And we'll talk more about that in a bit, but ultimately these nitrogenous bases make up the general structure of the nucleotide, which make up DNA. So purines, as we see on the left in green, let me highlight it in green. Oh, mouse, where are you going? What are you doing, mouse? There we go. Purines, guanine and adenine, they are double ringed structures. So those of you, again, who are familiar with grade 11 chemistry, it's got 2 carbon rings that make up those purines. While as pyrimidines in blue, are single ring structures, single ringed structures. So these single ring structures versus these double ring structures, it makes a difference in how it behaves. And we'll spend a bit of time talking about that as we move through this section. So DNA only contains the G, A, C, and T, or the guanine, adenine, cytosine, and thymine. Let me highlight these in blue just to kind of, bam, there we go. I'll erase these because we don't really need those. So uracil, as I highlighted in red, it's found in RNA strands and it replaces thymine. So when we talk about RNA, we'll spend some time talking about how uracil replaces thymine. These base pairs pair together in a very specific way, and this allows for the consistency of DNA to be maintained. I cannot stress how important this statement is right here, because without the way that it pairs, and I'll talk more about pure silver as well as the CT, but the key thing here with regards to how it works is that in order for us to see that consistency within all DNA for every living species, the DNA has to behave in a certain way. And I'll talk more about that in just a second. So I highlighted it up here and then I removed it, but I want to spend some time talking about it now. This statement, pure, oh, that's not what I wanted. There we go. Pure AG and then pyre CT. Pure silver helps me to remember the purines. Purines are A and G, which is silver. So pure silver helps me to remember that those are my purines. And then the other 2, C and T, are my pyrimidines. So this is just a little mnemonic to help you remember which of the nitrogenous bases go in which group. All right, questions? Questions. How pure AG relates to uracil? It doesn't. Don't worry about uracil right now. Because uracil is not a purine, right? It's a pyrimidine, but don't really worry about uracil right now. Why are there so many variations if it has the same purpose? Yeah, we'll talk a little bit about it in RNA. It's an evolutionary trait, ironically enough. But the reason that there's only 2 variations, right? The uracil and thymine are variations of each other. The rest are their own separate things. And the variations allow for that code to be robust. And I'll talk about the number of combinations and permutations that allow for DNA to have that robustness that allows for literally, right? Like when you think about the diversity of every single species that exists on this planet, it's because those 4 nucleotides are arranged in a special way for every single one of them, right? Every single one of them is arranged in a special way that allows for a bacteria to be so tiny and a complex elephant to be as beautiful and different from humans, from foxes, from other humans, from other foxes, from bacteria, from plant, like all of that is because those 4 nucleotides in some way, shape, or form can arrange themselves in different ways, right? And so it's those 4 variations that, even though it all has the same purpose, think of it like code, right? For those of you who've ever had to learn code, a 1 and a 0, it's just 2 variations. How on earth can it have so many different ways that it's expressed? And it's in that chain of that code. And it's that ability for it to be complementary. So let's take a look at that now. before I get too passionate about it without going into more detail about the things you need to know instead of the things that are just fascinating to think about. So again, the idea that these base pairs pair together in a very specific way allows for that robustness of variation and also containing or maintaining that consistency of DNA. So it is essential for the accuracy of DNA replication. DNA cannot replicate if there's no set code that allows for that pairing to exist. This specific pairing is known as complementary base pairing. Now, complementary base pairing revolves around the structure of those purines and pyrimidines. So when you think about the structure of those purines and those pyrimidines, let me highlight it actually, right? When you think about the structure of those purines and pyrimidines, in order for that bond to be formed with those nucleotides, because remember, DNA is a double helix structure, one side with one set of nucleotides, the other with another. They need to form a bond in some way, shape, or form to keep that double helical structure. And it forms a bond between A and T will always form a bond with each other. Okay, A and T will always form a bond with each other. They form a double bond through their hydrogens, and they are very strong attractions. And it is important for that bond to happen because it allows for that complementary base pairing, which allows for that variation. So A and T, they form a double bond together, whereas C and G form a triple bond together. Now, what do you notice? What do you notice about this bonding pair here, for example? Let's just look at A and T. What do you notice about that bonding pair? That's special, I guess, is a good way to say. What do you notice about it? Yeah, double hydrogen bond. Yeah. What else? There's one thing I'm specifically looking for, and let's see if anyone can see it. Oh, Emma, nailed it. Emma, say it in the general chat. You can just type it if you want. You don't have to actually get on mic and say it if you don't want to, but I mean, that's cool too if you want. Yeah, it's one purine and one pyrimidine, right? What do you notice about the C and G as well? Is it the same? One purine, one pyrimidine? I mean, obviously, because it's an exhaustive and there's only two other options, but it's one purine and one pyrimidine, okay? One purine and one pyrimidine. It will always form these bonds, AT-CG, right? AT-CG. So AT makes one pair, whether it's AT or TA, and then CG or KUGA, I call it, at KUGA to help me remember. The CG or GC, it will always form that bond. The AT or TA, it will always form that bond. It does not bond any other way, okay? Doesn't form any other way, not at all, not a chance. It won't do it. Okay, have I reiterated this enough to you all that this is the way that it bonds, and it is important that you know how it bonds. Okay, good. So no AC, correct. AC is not a thing. AG is not a thing. TC is not a thing. TG is not a thing. AT, CG, at KUGA, only and ever. That's how it rolls. Is AU a thing? Yes, I will talk about that later when we get to RNA, Matthew. I'll talk about that later. Alrighty, so now that we know how DNA forms those bonds, AT, CG, let me highlight the C and G. Okay, AT, CG. If we know the sequence of one strand of DNA, and this is where things start to get interesting, folks, because this is how cells think, right? If we know the sequence for one strand of DNA, we are able to predict the sequence of the other strand. So take a second take a look at this strand of DNA right here. And I want you to write that strand in, what the complementary base pair is. So I want you to think about that strand that I've highlighted in orange, or I've boxed in orange. And I want you to tell me what that complementary strand would be based on our understandings of complementary base pairing of our nucleotides. How would that second strand look if we know that first strand? Take a second, try it out, write it out, whatever floats the boat down the moat. And tell me what you think. All righty. Amina's got it. Yeah, the opposite side. Correct. The opposite side, Matthew. And then private message me afterwards, just so that people don't get spoiled. And then you can test it based off of what you finish and what I show you in a second. So Amina's got hers in. Thank you. Emma's got hers in. Okay. Sophia's got theirs in. Oh, okay. Only 3 of y'all trying? Or you're just thinking about it, some of you still? Colin's got theirs in. Oh, Colin. Oh, Colin. Where's your house? Minus 10 points to your house. Minus 10 points to Colin's house. Hold on. Let me pull it up. Let me pull it up. Thank you, Olivia. Yeah, where's Colin's house? Colin's house. Oh, Colin's house. House, I'm not going to say that long, or city in Wales or whatever. Minus 10 points to House Ilian, blah, blah. I can't pronounce it. It's too long. Shame. Yeah, shame. I am sorry. It's okay, Colin. I don't mind. It's just your house members that might mind. It's fine. It's fine. It's not a big deal. It's not like you were in second place and you're competing for the House Cup with House Furry Alien. It's fine. Whatever. It's not a big deal. Not a big deal. Not a big deal at all. Alrighty, a couple of you have sent it in. My hope is that the others who have not sent it in, you're at least trying to think about it. Maybe I'll give you all 1 more minute. Can we have the 10 points back if he's right? No? Nice try. Is he right, though? Let's see. I don't know. We'll see. Nope. Alrighty, let's take a look at what it is. And you tell me if you got it. For those of you who didn't send it in, that's okay. For those of you that did, thank you very much. So if you made a mistake, think about where you made your mistake and think about why you made your mistake, and then ask questions if you have it. So those of you who didn't send it in, that's okay. My hope is that you got the gist of it. I'll tell you right now, right off the bat, folks. You're gonna have a question like this on your quiz, right? You're gonna have a question like this on your quiz. I will give you a strand or a sequence of DNA, and you will tell me what the complementary base pair is. The diagram is 3D. Yes, you're talking about this diagram here, right, Matthew? Yeah, that's 3D. It's just hard to express 3D because I'm not good at drawing outside of like what you see in my drawings. They don't actually touch. No, it's supposed to look more like this. Let me see if I can replicate it. It's really hard to draw in 3 dimensions. And then it kind of comes up and under and around, doesn't touch. Yeah, it's kind of like that. Sir, what do you mean you're like the best artist? Ha, ha. Yeah, that's how it looks. Yeah, exactly. It looks like a worm. Yeah, Picasso over here. Hey, easy now, easy now. Yeah, the bonds are the lines in the middle. So I'll draw them in color here just to kind of show. Bam, they kind of form that bond along here like a ladder. I'd add shadows in for depth, but You get the gist of it. Now it's a snake. Yeah, now it's a snake. Now it's really a snake. Hold on, let me just complete the snake. Oh, wait, hold on. There, now it's a snake. Now it's a snake. Snakes and ladders, yeah. Anyways, DNA, it's a snake. No, it's not actually a snake, but hopefully you get the gist of it. I'll share a video maybe in the outline just to kind of go over, thank you, the three-dimensional structure of it because it helps you to visualize it a little bit. Normally I have this thing that I pass around and it's this folding double helical wooden structure, and you can kind of get a feel and look for how it is. But I digress in the sense that I cannot send this to each of your individual homes. So is what it is. Thank you. I appreciate the kind words about my art skills. Appreciate it. You're all just fishing for house points at this point, as far as I'm concerned. And then, is it working? Well, who knows? All righty. Are there any more questions with regards to DNA in terms of sequencing? Because we have another sequencing question that I want you to try. Okay, I have another sequencing question that I want you to try. One second. All right. Oh, question. No, okay. So let's take a look at one more sequence. And I want you to think and wrap your head around the idea as I share my screen with you. And I want you to think about what if there was an error in the DNA sequence? What if there was an error in the DNA sequence? And this time you do not have to write it in because I want you to make a mistake and I will share that mistake with you. But imagine there was an error, okay? And I erased it the wrong way, but whatever, it's too late now. I wanted you to find the error. But here it is. Here's our error. What is the mistake here? What is the mistake? What is the mistake here? Yeah, it should be a T. Exactly. It should be a T, right? That base pair, this A was mistakenly placed in there when it should be a T. So there's two A's across from each other. That causes the helix to be the wrong shape and the wrong size. And as a result of that, it is what's called an error. So the cell will check the DNA hundreds of thousands of times per second to prevent those mutations from being copied or causing a cell to be damaged. Now, we've talked about mutations a little bit so far. We will spend way more time talking about it in next unit, but these mutations are bad, usually, okay? But again, when you think about our cells, right, just our skin cells alone, okay? Just our skin cells alone. Just our skin cells alone. How often, who knows this? I'm curious to see who knows this. How often do your skin cells replicate and create a new layer of skin? How often does that happen? I've talked about it once before. Who kind of remembers or kind of knows this? How often does around 30 days, 2.5 to 3 weeks it takes. So your skin that you have now, right? Look at it. Look at your skin, okay? The skin that you have now, in about 3 weeks' time, will be completely brand new cells. Completely. All of them. Every single last one of them. Okay? Every single last one of your skin cells that you see on your body will be completely replaced in the span of about a month. Now, each cell has to copy itself in order for that to happen. And in order for it to copy itself, it has to copy its DNA. And in order to copy its DNA, it goes through 46 chromosomes, makes a complete copy of each of them. Hundreds of millions, if not billions of DNA nucleotides need to be perfectly copied in order for that cell to replicate. Once in a while, a mutation will happen. I don't shed. What happens? Yeah, it's a lot of work. Everyone sheds skin. You may just not see it. Yeah, it's a lot of work, right? So Isn't that dust? Yes, dust is just dead skin that has dried out. And yeah, it's dust. I got dust on my computer. Here, look, dust. That's old Mr. Q. You literally die and are reborn every once in a while. I don't know if you can see it. I gotta see. Hold on. Let me turn on my camera for myself. Let's see. I don't know if you can see the dust. Yeah, you can see the dust. Bam. That's old Mr. Q. Skin cell that is desiccated. If you accidentally inhaled dust, does that make you a cannibalism? Great question, Amina. No, because the skin is desiccated. It's not actual flesh. Cannibalism is defined as flesh, and skin is not flesh, technically. Technically. It's the meat. That's what makes you a cannibalism. Or a cannibal, I should say. Ooh, this turned dark. 10 points to Amina's house for turning the conversation to the dark place that is cannibalism. 10 points, 10 points. 10 points to Amina's house. Dark question, but I appreciate it. I appreciate it. I'm pulling the weight around here. So, with regards to those mutations, right? Again, your skin cells replicate one time fully in the course of a month. Hundreds of billions of cells have to replicate themselves. Hundreds of billions of DNA nucleotides need to be copied. So once in a while, right? It's not a perfect mechanism, but once in a while it makes a mistake. And the cell will attempt to fix these errors. If there is, however, extensive damage and the cell is unable to fix it, it will go through what's called apoptosis. Now, you may remember apoptosis from the virus lesson that we had last unit. The specific instructions that happen that tell the cell to go through apoptosis if there's a mutation or an error with that DNA replication, that's the same process that the virus activates when it's like done and dusted replicating inside the cell and it wants to reach its next targets. It triggers that specific sequence and then it's time to go. How fast do they replicate? Great question. I'm going to show you this video because this is, this video, let me pull it up on my main and then I'll bring it over. DNA replication 3D model. Is this the one? Hold on. Oh, this is the one. Beauty. Okay. Here we go. Do mutations occur often? I don't remember. Can you go over that again? It's not important, Matthew. It's just the idea that cell death or apoptosis happens if a cell has a mutation that it cannot fix. Jessica, I'm going to show a video in a second that talks about DNA replication. It is a super cool video. I'll post the link in the chat after I show the part that I want to send. But let me finish this lesson first, and then I'll show that video. Do mutations occur often? Oh, yeah. I would say cells are about 97% accurate with their DNA copies, right? So 3% of its copies have an error. We'll talk a little bit about error correction because the cell is also very good at correcting those mutations, not just through apoptosis or cell death, but it can remove that error and fix that DNA if there is an error. But I'd say it's about every 1 to 3% there's a mutation that happens. Of those mutations that happen, only about 1% make its way on to replicating. The cell is like 99.9% accurate. Is cancer a mutation? Yes. Cancer is a form of a mutation that causes the cell to replicate uncontrollably. So Colin, to answer that question, what happens when it mutates? Several different things can happen. Sometimes nothing. Sometimes it's a birthmark. Sometimes it's a birthmark. That's technically a mutation. I'm a mutant. Anyone who's got a birthmark, you're a mutant. Yeah, our cells are so smart. It's unreal how smart they are. They're very, 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 very clever. I'm surprised. I feel like more mutations would be so much more common. Well, again, Ava, right? When you think about it, if only 1% of that mutation happens, 1% of 17 billion is still a lot, right? It's still a lot. 1% of a billion is like 1,000 or whatever. Or 0.1% of a billion is 1,000, right? So it's still a lot. Only if my brain feels. No, it has nothing to do with. No, well, there's a 3% chance of a mutation. There's a 1% chance that that mutation gets passed on. And of that 1%, depending on your familial history and depending on all the other things, there's different chances for it to actually become cancerous, right? It's a percentage of a percentage of a percentage, just randomly, right? Just randomly. Now, there's other things that come into play here, like familial history, exposure to radiation, exposure to cancer-causing agents. So it's not a cut and dry, everyone has a 1% or a 2% chance of cancer. But that is what factors into it, I guess, is what I'm saying. Okay, let's move on to the last part with regards to packaging of DNA, so I can show you this super cool video afterwards. Oops. And talk a little bit about DNA replication, which we will spend more time talking about tomorrow and the day after, as well as throughout the course of this unit. So packaging DNA. If you were to take all 46 chromosomes or all of our DNA from just one single cell, the DNA would be approximately 2 meters long. So if you're 6 feet tall or you have a meter stick kicking around, double that, and that's the approximate length of one cell's DNA. So one single skin cell has DNA that's approximately 2 meters long. So this DNA needs to fit inside of a microscopic organism or a microscopic cell. And it does that by tightly packaging and winding it up into our chromosomes. Question. How is it taller than me? Hey, it's taller than me too, Jessica. Don't feel too bad. It's taller than me. It's taller than most people in this class, I would suspect. I don't know how many people are over 6 feet tall in this class, but it's taller than most people in this class, I would suspect. Your dad's 6 feet tall, 6'6". Yeah. So it's not taller than Emma's dad. Yeah, well, my dad's 5'6". In your face, Emma. In your face, Emma. Bam. Okay, so almost double that. Yes. Well, no, 6 feet tall is about 2 meters. So it's approximately Matthew's. Yeah. Matthew's dad is about 6 feet. Oh, isn't 6 feet like 1.8 meters? No, 3 feet is 1 meter. 6 feet, it's like 197. Hold on, let me double check. How many meters is 6 feet? 1.82. You're right, Matthew. 1.82. So a bit taller than 6 feet. So yeah, about Emma's dad's height, I guess. So Emma, when you go see your dad later today or whenever you see him, say that, no, not your dad. Not your dad, Matthew. Emma's dad, sorry. Yeah, say like a DNA from one single strand is about the height of Emma's dad. That is the new unit of measurement for a DNA length. So how does it get packed into that tiny, tiny single cell? Well, we have what's called histones. These histones are able to wind the DNA up like a string or a spool, and it winds up the DNA nice and tight and compact without damaging it. And many, 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 many histones are needed in order to wrap up that 2 meters worth of DNA. And then those histone wrapped DNA sequences coil up and they form what's called a chromosome. It is very, very compact, and the cell will open and unwind different sections of that DNA as needed for replication purposes, for protein purposes, or anything and everything in between. All righty, questions? Let me get this link for this video that I want to show because it's dope and I want to share it with you all. Oh. And hopefully that works. I can't see my monitor right now. Get out of here. Bam. All righty. And then if anyone has questions about any of this content, let me share my share computer screen, audio. Alrighty. This lesson will be put in the recording folder later. Yes, it sure will. It sure will. Yeah, it's a lot of content, but it's a good intro because it's going to be based off of the next couple of lessons. So our understanding of it will definitely help in as we move throughout this unit. Alrighty. Can you see the screen now? I know there's probably a big gray box that's moving right now, right? Is it moving when I move it? Oh, 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 no box. Okay, beauty. Okay, you can see it. Can you hear it? Can you hear the weird alien sounds? Okay, beauty. So this is a really super cool video. I first experienced this video. How old is this video? Oh, this is, wait, I hope this is the right one. Whatever, it's fine if it's not. I'll find a equivalent if it's not. But I first experienced this insane video when I was maybe a bit older than you all, like 19, 20-ish. This video is very old, and it looks like yarn crystal cavern. Yeah, it is very old, but it is a great three-dimensional replication of some of the processes within the nucleus. So let's watch it and experience the joy that it is, and how crazy it is that this happens. So there's a human cell 10,000 times. Pretty neat. All right, here's the good part. That's DNA. And grapes. So what do you think those are? What do you think it's what do you think those are, those purple things? We just looked at them. If it's organizing your DNA, trying to coil it up, what coils up that DNA? Histones. Ding, 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 ding. Good job, Colin. All right, let's watch it coil up. So look at how it tightly packs up all that DNA. All of that is DNA. One single chromosome. Bam. And now that cell's dividing and getting set. We'll look at this video again tomorrow when we talk about mitosis. But and there it goes, splitting apart. One cell, two cell. Cytokinesis, kablam. So we'll get to the part that blows my mind every single time. So Jessica, you asked this earlier, how fast? So yeah, 2003. This is, yeah, I saw this video 2003. Oh, heavens. Some of these terms sound like wizard spells. They are kind of like wizard spells. So in 2003, this video came out. Oh my God. And I saw this video and it blew my mind because when you think about, this video is older than, yes, it is older than most of you. I forget that adults now also had the internet when we were young. Yeah, hey, Amina, I got my first internet-ready computer when I was 14 years old. So not much older or not much younger than you, but it was one of the coolest things that ever happened. I played StarCraft 1 through dial-up internet. If you don't know what dial-up internet is, oh my God, that makes me feel ancient. But that's okay. The OG, yeah, the OG StarCraft and the OG internet. But I first saw this video in 2003. To talk about the speed, that DNA replicates at. Just watch how quick this process happens. And I'll liken it to, if the video doesn't do it, I'll liken it to the speed of something later. That's a whole adult person to go. Yeah, Amina knows what dial-up is. Okay, so here it goes. Billions of these machines copying DNA. Look at it go. Pew. Look at how fast it goes. This is real-time speed. This happens every single time your cell needs to replicate. This is one of those processes of interphase that we talked about, or S phase with regards to synthesis. You don't need to see this part. But that's how fast it goes. It looks like a conveyor belt. Yeah, it's definitely some factorial stuff. But it goes the speed of a jet engine. So a jet engine, I think, goes somewhere up to Mach 1 to Mach 3, depending on the type of jet engine. But a jet engine can go anywhere from 600 to 800 kilometers an hour. So that's if it were traveling or if it were going, we measured in terms of RPM and rotations, and it's equivalent to a jet engine. And that happens in every single cell, every single time it replicates, hundreds of millions of times a day in hundreds of millions of cells a day. Genetics. It's going to be a great unit, folks. There's about 15 minutes left of class. Please take a look at your homework. You have to sign up for Explore Learning. You have a couple of practice problems to review your content. Great first lesson, folks. Great first lesson. If you got questions, you're welcome to stick around. I'll be here until 10:20. But this is part of our asynchronous part now. What did you share, Neva? What's this video? Let's see. Why is your body amazing? Yeah. Great video. I know this. I've seen this video. ASAP Science is great. Yeah, watch it. It's fantastic. The human body, any living organism overall. Folks, this is why biology to me is the most fascinating study and the most fascinating field. It's just, you can't compare it to anything else. You can't compare it. Cannot compare. All right. So take a look at your outline, folks. You have a couple of things that you need to do for homework. Sign up for Explore Learning and also do a couple of questions from the textbook. You have about 15 minutes left of asynchronous class. If you want to head out of the Zoom, you're more than welcome to. If you have questions, you want to stick around and ask them, I'll be here for another 15 minutes or so. All right, folks. Sir, can you please share the video link? Oh, yeah, sure, the video link. Sure thing. Wait, the video link for the thing I just showed or the video link for the lesson? You're welcome, folks. See you later. Explore Learning, Matthew. Click the link. It's in our outline. Yeah, the YouTube one. Here, I'll share the link for the YouTube one. It's going to be in the outline. So I'll put cell processes video. Yeah, there. It's in the outline for today's lesson. Fan. Yep. See you later, folks. Bye, everyone. No problem, Matthew. Yeah, click the link. Uh-oh, what did I do? I made a mistake. Sir, what's pure AG? I cannot understand. So let me get my whiteboard up for you here. So purine is equal to pure. Oh. Okay. Pure AG is A and G, or adenine and guanine, are purines. That's all that means. It's just a little mnemonic way to remember which one is which and which group they're a part of. Does that make a bit more sense, Fan? What's this? A kind of cell or something? No, this is just to remember. It's like a quick little way to remember like the nucleotides, because adenine is A and guanine is G, right? So adenine and guanine are purines, right? Adenine and guanine are purines, and pure AG helps us to remember that they're purines because purine starts with PUR, and pure AG is just a quick way to remember that adenine and guanine are purines. Yes. Cool. No worries. See you later. Cole, Mariam. Oh, Matthew, yeah,
Mr.
what's up?
Kidasol, have a great day.
You too, Matthew. Take care, bud.
Mr. Kitaso,
Oops, question. I can't figure out this question. When is the production of genetically identical offspring a disadvantage? Yeah, you got to think a little bit about that. In terms of a disadvantage, think about. Yeah, you got to think about that one. I won't spoil that one for you. I'll just say think about why genetically identical things, if, for example, a human, right? If a human is prone to sickness and it were making a perfect clone of itself, is that a good thing or a bad thing?
bad.
Okay, so there you go. There's a disadvantage, right?
Yes. Okay, thank you.
No problem, Fei Yang.